Hi everyone, welcome back to neetpg. This video is about the general embryology part. So in this video, we'll talk about the first week of development, the processes and events happening in the first week of development. So let's talk about fertilization. So in the first week, fertilization takes place, and it takes place in the ampulla part of the fallopian tube. So there is a zona pellucida, a membrane which is preventing the implantation and polyspermy. So in order for the fertilization to take place, the sperm have to get inside the zona pellucida, or sperm have to bind to zona pellucida, and there is a reaction taking place. So if once the sperm bind to zona pellucida, it triggers a release of the acrosomal enzymes from the sperm, so which is called acrosomal reaction. So this acrosomal enzymes are like, it contains three enzymes, which is acrosin, acid phosphatase, and hyaluronidase. So these three enzymes contains in the acrosomal reaction, and once these enzymes are released by through this acrosomal reaction, sperm is able to penetrate the zona pellucida. And once the sperm penetrates the zona pellucida, there is cortical reaction. It means that in the cortex, or the cortical granules are there, and it releases its secretion. So that process is called cortical reaction. So thus by releasing those secretions, what happens, the cortical enzyme will reach the zona pellucida layer, and thereby changes the permeability of the zona to sperms. So, and this reaction is called zona reaction. So thereby what happens now, this zona is not will not allow the entry of sperm. So thereby, because of this reaction, we are able to not allow the polyspermy occurring, because now the zona is impermeable to sperm. So afterwards, so so first of all, there was acrosomal reaction, then there was cortical granules releasing their secretion. So there was cortical reaction, and then there was zona reaction. So these were the process of reactions which were taking place, and then the sperm has entered the zona pellucida, and now there is fusion of the male and female pronucleus, and because of the fusion we have the zygote formation. So the zygote is formed, and now there are events taking place in the zygote. So this zygote, it undergoes the first cleavage, and first cleavage afterwards, then we have this 2-cell stage, then the second cleavage taking place leading to 4-cell stage, and the third cleavage causes the 8-cell stage. So these up to 8-cell stage, these cells are like loosely arranged, but afterwards, after 8-cell stage, after 8-cell stage, combustion takes place. So after 8-cell stage, the cells are like held together by tight junction, and this process is called combustion. So up to 8-cell stage, it was loosely arranged, and after 8-cell stage, cells become tightly arranged because of tight junctions. So basically, after 8-cell stages, there is difference. Then afterwards, then there is differentiation. So and it becomes 16-cell stage, and it is called morula. And in morula, there is cavitation taking place, and because of cavity formation inside the morula, then we have the blastocyst. So, and after cavitation, we have the blastocyst, and afterwards in blastocyst, a process called hatching takes place. It means that the zona pellucida layer, which we talked about, becomes disappear, and then because the zona pellucida is something which is preventing the implantation and preventing the polyspermy, now the zona pellucida has disappeared, then we have the implantation. Okay. So in this diagram, we can see the zona pellucida layer, which is a green color. Okay, then we have a process of reactions, which I said, like first of all, when the sperm comes, there is acrosomal reaction, Then there is granules release the secretion, and thereby they changes the permeability of zona, which is zona reaction. And once after the zona reaction, this zona becomes impermeable to sperm. There will not be any entry of other sperms. So it is like zona reaction causes the polyspermy blocking. So once after hatching, once the zona pellucida layer disappear, then this will get implanted. Okay, then now I'll talk about the time and site of events. So this video is about first week of development. So we'll see like first day what happened, second day of first what happened after fertilization. So we'll look into the site of events. So fertilization takes place like within 12 to 24 hours of ovulation, and I guess you saw the first video of gametogenesis. In that I explained like why fertilization has to take place within 12 to 24 hours of ovulation, because the secondary oocyte in the metaphase arrest will wait only for 24 hours. So within that time period the fertilization has to take place. So like within 12 to 24 hours of ovulation the fertilization takes place. So after the fertilization, one day after, it will be like 2 cell stage will be formed, and the third day after fertilization 8 to 16 cell stage will be formed, and we know that 16 cell stage is called morula. So third day after fertilization morula will be formed, and the fourth day after fertilization 32 cells will be formed, and this 32 cells is called advanced morula, and this advanced morula will be entering the uterine cavity. So the advanced morula, which is 32 cell stage, enters a uterine cavity. So once it enters a uterine cavity, basically the uterine fluid will enter the advanced morula, and thus when fluid enters this advanced morula, then blastocyst is formed. And at the time of formation, so the blastocyst will be containing like 58 cells. So the 53 cells will be in the outer cell mass, which is forming the trophoblast, and the trophoblast, tropho means nutrition, so like trophoblast will form the future placenta, and the 5 cells out of this 58 cells, 53 was in the outer cell mass, which forms a trophoblast and which forms a future placenta, and the 5 cells will be in the inner cell mass, which forms a embryoblast, and this embryoblast will form the future embryo proper. So this blastocyst, when the advanced morula enters uterine cavity, this blastocyst will be formed on the 4.5 day after fertilization, and the 5th day after fertilization, as I said, the process of hatching takes place and zona pellucida layer disappears. So by 6th to 7th day after fertilization the implantation will be happening. So this is a diagram. So in this picture we can see the series of events taking place, like in 12 to 24 hours of ovulation, fertilization taking place, then after 24 hours or like after 30 hours there is 2 cell stage, that is after 1 day of ovulation, and the third 1 day after fertilization, and the third day after fertilization like 16 cell stage will be formed, which is morula, and the fourth day after fertilization we have the advanced morula, which is 32 cell stage, and 4.5 days zona pellucida layer will disappears, and by 6th to 7th day fertilization, after fertilization implantation will take place. So this is a blastocyst. So blastocyst contains the inner cell mass, which is embryoblast, and the trophoblast, which will form the placenta, and also the cavity cyst means cavity because of the uterine fluid entering, there is cavity formation, which is called blastocele. So here we can see the blastocyst have inner cell mass, which is forming the embryonic stem cells and which forms a future embryo, and the outer cell mass forming the placenta. So that was all about the first week of development. So now an applied of understanding of first week of development. There is ectopic pregnancy, where the what happens is that the fertilization takes place in the ampulla, but the implantation should supposed to be takes place inside the uterine cavity, the endometrial layer. But here the implantation is taking place. Most common site of ectopic pregnancy will be ectopic tubal pregnancy, means the implantation is taking place in the fallopian tube. So, because the implantation is taking place in the fallopian tube, the trophoblast, which is the outer cell mass, then will further divide into syncytiotrophoblasts, and this layer will not be well developed. And this is a layer, syncytiotrophoblast is the thing which will be producing HCG levels. And because here the implantation is taking place in the fallopian tube, syncytiotrophoblaст layer will not be well developed, thereby the HCG levels will be very low. So in order to diagnose a case of ectopic pregnancy, first of all, the female should be pregnancy positive. If we take the urine test, should be pregnancy positive. And if we look into the serum HCG levels, it will be like very low, because of this syncytiotrophoblast is not well developed, so the HCG levels are very low in the body. Usually the HCG levels will be like doubling. So ectopic pregnancy condition will have a clinical feature similar to appendicitis, means the female will come with vomiting, abdomen tenderness, pain, and all. So the features will be similar to appendicitis. So what is the treatment of this case? Treatment, of course, will not tell the patient that congratulations, she is pregnant. Well, it's ectopic pregnancy. So the treatment should be the termination of pregnancy, because if we don't terminate the pregnancy as soon as possible, if we get to know it is ectopic pregnancy, then there is a complication of tubal rupture if this case is not treated. So once we got to know it is ectopic pregnancy, the treatment is immediate termination of pregnancy. So if a patient, it's very important that if a female patient comes to us and telling the symptoms of appendicitis and the clinical features of that and all, it's very important to ask the menstrual history of the patient. And if the patient is telling like she had no periods in the previous months, or she had amenorrhea, it is important to look for the urine pregnancy test and the serum HCG levels, because it can be a case of ectopic pregnancy as well. So that's all about this video. Thanks for listening, and please like, share, and subscribe the channel. Thank you.